Okay, so I put a little gray box in the upper left-hand corner. If you want to move the video up there, then I think you'll be able to see all of my slides. Today I am talking about a project looking at pyrethroid resistance in Culex tarsalis. And this project was primarily completed by two of my graduate students, Emiko de la Vega and Bonnie Ryan. Before I get started, I want to thank my vector control collaborators. So projects like this are definitely not possible without them. San Joaquin, Saciolo, Placer, and Lake County, all in California, and then the Salt Lake City district in Utah. I worked with them to develop the project. They helped with collecting mosquitoes. For this project, a lot of the bottle bioassays were done at the districts, and they also provided funding. In addition to the funding from the districts, this project is also funded by PACVEC and the University of the Pacific Graduate Program. So I'm going to be talking about Culex tarsalis, probably not news to anyone here. This is an important vector of West Nile virus and other arboviruses in the Western US. Historically, this mosquito is a rural mosquito, though now it is often found in residential areas as well. Because of where it's found, it faces insecticide pressure from agricultural spraying as well as vector control, and until recently, few reports of insecticide resistance had been reported. Now, I will say that over the last few years, that has changed. There have been more and more reports of resistance in tarsalis. A couple of goals for this project. The first one was to determine the prevalence of resistance to specifically pyrethroids in select study sites across Northern California. Then after establishing that there was resistance, we wanted to look at the mechanism. So what's happening in the mosquitoes that can lead to this resistance. We looked both at enzymatic resistance using enzyme assays that happen in microplates. Sometimes they're called microplate assays, and also looking for target site mutations. So to think about the resistance mechanisms, it's important to know how the insecticide works. So here's what is likely a review for you for how pyrethroids work. They target the nervous system of the mosquito. So first, that star there represents where the action happens. The pyrethroid would bind to voltage-gated sodium channels. This binding actually holds those channels open. This increases the influx of sodium ions into the neuron. That increase of sodium increases the number of action potentials, which increases the amount of acetylcholine that is released from the cell. That acetylcholine goes to other neurons and muscles to overexcite them. So basically with this overexcitation, the mosquito uses all of its energy sending action potentials and contracting muscles, and that leads to the mosquito's death. So mechanisms of resistance that combat or that prevent pyrethroids from working. The first one that I mentioned, and I'll say a little bit more about here, are detoxifying enzymes. So this is when mosquitoes actually produce more enzyme that can break down or inhibit the insecticides. This diagram is a little complicated and it shows information for all types of pesticides, or at least for many types of pesticides, but I want you to just to focus on pyrethroids since that's what I'm focused on here. And the takeaway is that the enzymes known as esterases, oxidases, and GSTs have all been shown to lead to resistance for pyrethroids. The second type of resistance mechanism that we looked at are target site mutations. Target site mutations are actually a mutation in the DNA itself. This mutation in the DNA leads to a change in the amino acid and the protein that keeps the insecticide from binding to its target site. For pyrethroids, the mutation of interest is something called KDR, which stands for knockdown resistance. This mutation prevents the pyrethroids from binding to the voltage-gated sodium channels. And there are two common mutations. There have been others discovered, but these are kind of the starting point when looking for resistance. And that is a change from the amino acid leucine to phenylalanine, or from leucine to serine at one place in the gene that codes for the voltage-gated sodium channel. So Let me dive into these goals. So first, looking at determining the prevalence of resistance. And for this, we did CDC bottle bioassays. This work happened in the summer of 2018. First, mixed age females were collected using CO2 traps. We had about 3 to 5 populations per district, and there are those California districts that helped me out greatly with this project. For the bottle bioassays themselves, we had kind of 4 categories of bottles. So the control that didn't have any chemical, right? Acetone only, that acetone evaporates, leaving just the clean bottle. We had PBO only, PBO being a synergist that inhibits oxidases, one of those enzyme groups that I talked about. Permethrin only, and then permethrin plus PBO. For each of those groups, we ran these in triplicate, so 3 bottles, and about 20 mosquitoes in each bottle. Every time the bottle bioassays were run, we ran a susceptible colony so that we could make sure that the assay was working like we wanted to, along with the wild-caught population. And in most cases, we were able to do 2 biological replicates. That is, we were able to repeat it twice for the field site. After the mosquitoes were in the bottle, we recorded mortality every 15 minutes for 2 hours. Here is a graph that shows the type of data that we get from the bottle bioassays. You perhaps have seen these before. On the y-axis, we have mortality. On the x-axis, we have time. So again, with those 15-minute checks to see whether or not mosquitoes had been knocked down or were dead. The green lines here are for our susceptible colony. You can see that they died early on in the experiment, which is what we want, what we expect. They died in about 15 to 30 minutes. The orange lines here are for the wild population, in this case, Empire from San Joaquin County. And if we look at the permethrin only bottle for this particular population, we can see that mortality neared 100%, but it didn't quite reach it. However, when PBO was added, mortality happened much faster. So in this case, mortality reached 100% at 45 minutes. An example from another population, this is Winters in Yolo County. Again, we have our susceptible colony in green. Now this is a different colony. The summer we did this, it was actually quite difficult to get colony mosquitoes. A lot of districts, their colonies were down that summer. So we used whatever colony we could find. It's not ideal, but it's what we could make happen for that summer. You can see this KNWR colony took a little longer to die, but still everything had died by 60 minutes and most of the mosquitoes by 45 minutes. Now looking at the wild population, only 8.6% of the mosquitoes died by the end of the 2-hour point. So quite a lot of resistance in this population. When we added PBO, you can see mortality increases, but in this case, it still never reached 100% by the end of the 2 hours. Now, for the sake of time today and for the sake of everybody's sanity, I'm not going to show you 15 more of these, but we do have the data for 15 other sites. Here they are summarized in this table. So on the very left, you have the district in California. Then we have the column that shows the specific site. These are percent mortalities. So if we look at percent mortality for permethrin only, a main takeaway here is you can see there's a range, right? So there was variation from one population to another. The highest mortality, 96.2%, the lowest mortality, 1.5%. All right, then if we look at the numbers for permethrin plus PBO, so here we've added the synergist that helps to inhibit those oxidases. Good news here, mortality increased in all cases, and in almost all cases, mortality went above 90%. One exception here, that Winters population that I already showed you, where mortality did not move over 90%, even with the addition of PBO. Okay, so from the first goal, we found, yes, we do have resistance. In fact, it was probably a bit more than we expected. So next steps then were look to see what was happening in the mosquitoes that was leading to that resistance. First step, the enzyme testing, looking for enzymatic resistance. So to do this, the mosquitoes get ground up, and that ground up mosquito or the homogenate gets added to these plates in triplicate. The enzymes, those detoxifying enzymes that we're trying to measure, are found in that mosquito homogenate. Then you add a substrate or other reactants to the plate. This starts a chemical reaction that produces a product that has a color. So depending on how much enzyme activity there is, you get kind of a darker or a lighter color, and that can be measured using an instrument that measures the absorbance. So it measures how much color or how much product is in that plate. We tested for these groups of enzymes: oxidases, alpha esterases, beta esterases, GSTs, and acetylcholinesterase. We also did a general test for how much protein was there, and that helped us normalize for the size of the mosquito. So these box plots are how we show the data from those enzyme assays. Let me just take a minute to walk through this first one. So on the y-axis, we have the amount or the activity of the enzyme. In this case, we're looking at alpha esterase. It's divided by the amount of protein to help normalize for the mosquito size. Across the x-axis, we have different populations. BFS on the left is our colony, and then this is some of the San Joaquin County locations on this particular graph. For the box plot, in the box itself, basically that represents the amount of enzyme for 50% of the population. If you go all the way to the whiskers, that takes in most of the population, and then you'll see some dots above those whiskers. Those are outliers, so mosquitoes that are expressing a lot of these particular enzymes. One more thing to point out on this graph are those letters. Those letters denote statistical significance. So if a particular box has a different letter, then it may be statistically significant from the others. So specifically about alpha esterases, the first thing you'll notice is that our colony levels are high. Not at all what we expected and not at all good news. Basically what that means is that our susceptible colony that we had hoped to use as a reference actually had much higher levels of esterases than we expected. You can see some trends from the populations here. So Empire and Vine, for example, have higher levels of alpha esterase than the other populations shown. We see a similar thing when we look at the beta esterases. Not too much difference in the wild populations, though Vine is maybe a bit higher than some. The BFS colony, though, by far the highest. Again, this is not what we expected and really leads to the unfortunate takeaway that our colony is not a good comparison colony when looking at this data. Moving on to oxidases, now we expected oxidase levels to be high, and we know that oxidases are at play because of the bottle bioassay when we added the PBO. We didn't quite see that, but again, we're struggling with how to interpret this data because of the issues with the colony. So putting that to the side, we can see a couple of populations where the mosquitoes had higher levels of oxidase, namely Vine and Empire. Similar trend to oxidase for the acetylcholinesterase. We can see that Vine population again has quite high activity for acetylcholinesterase. All right, so without having a colony to compare to, that's how we had planned to do all of our statistical analysis. But because of what we found, that type of analysis isn't going to work for us. So I'm currently working with a statistician to figure out different ways that we might analyze this data to see what information we can take from it. One thing we're doing is looking at regressions. So here are some really preliminary regressions. So I wouldn't want you to leave saying this is absolutely the truth, this is what's happening, but this is a work in progress. So if we look at these regressions, we have survivorship on the y-axis. So higher on the y-axis means more mosquitoes survived, or there's more resistance from those bottle bioassays. And then on the x-axis, we have enzyme levels, or a measure of enzyme levels. What we can see is for acetylcholinesterase and the GSTs so far, there is a significantly positive correlation. So as those enzyme levels go up, so too does survivorship. And it's near positive for oxidase. So we are working to add more populations to this analysis so that we can get kind of a more robust or a better feel for how these enzymes might specifically be affecting resistance. Moving now to the target site mutation. So this again is that DNA mutation. So for this we have to do molecular assays. It's specifically a cybergreen qPCR. And we have 3 different primers that match up to the 3 different alleles we're looking for, that leucine, phenylalanine, and serine. The underlined letter here is the DNA mutation that distinguishes those 3 alleles. And then we add those tags that you can see in green. That basically just helps us differentiate them. I'm not going to go into the details of how this assay works, but here's what the output looks like. So the takeaway for you here is that based on where that peak is, we can tell if it's phenylalanine, if it's leucine, or if it's serine. For those of you who may be interested in this assay, right now it can't differentiate a leucine serine from a serine serine, but that's a pretty rare combination, and we do have a secondary test that can help with that. I'm just going to show you a bit of the data that results from this assay. So here are some of the populations from Lake Placer and Sac-Yolo. On the left, we have that KNWR colony. This is our susceptible colony. Good news, all of the alleles in that colony are the leucine or the wild type or the susceptible allele. So the green here, leucine, is our susceptible allele. So that's what we expect. That's good news. Not so great news when we look at the rest of the population. So for each population, you can see a small green bar at the top usually, or maybe not a green bar at all. That's the susceptible allele. Then we have serine and phenylalanine shown in the kind of yellow and red color. Those are our KDR, our resistant alleles. So the main takeaway here is that in many of our populations, and most of our populations, those KDR alleles are more prevalent than the susceptible allele. So while my grad students were working on this project, I was also teaching a class, and I was looking for a project for those students to do. And I thought, what data do I have, or what mosquitoes do I have that I might be able to look to see how long KDR has been prevalent? So I dug into the freezer, I found some Culex tarsalis that I had from my days in grad school, and I was able to pair up or closely pair up a couple of sites. So in this slide, on the left-hand side are samples that were collected in 2006 to 2008. On the right-hand side, samples collected 2016 to 2018. 2 sites are nearby sites in Yolo County and nearby sites in Sutter Yuba County. The takeaway here is that 10 years ago, 12 years ago, give or take, there were far more susceptible alleles in our Culex tarsalis populations than there are today. So to summarize, there is substantial resistance in pyrethroid to pyrethroids in Culex tarsalis in our study counties. Mortality ranged from 1.5 to 96.2% in those bottle bioassays, and the addition of PBO brought mortality above 90% at most sites. The enzyme data is a little cloudy because we don't have a good colony to use as a reference at the moment. However, based on those regressions, there's a potential that increased oxidases, GSTs, and acetylcholinesterase might be leading to resistance. Finally, we have a high prevalence of those KDR alleles, particularly the leucine to phenylalanine mutation. There are some serine mutations as well. And the prevalence of these alleles has greatly increased in the last 10 years. With that, I say thank you and take questions. And also, if you think you might have susceptible wild tarsalis, I would absolutely appreciate you reaching out, because we're hoping to find a new susceptible colony.